Next, we're going to hear from Dr. Ted Price, PhD. He's professor in the School of Behavioral and Brain Sciences at the University of Texas in Dallas since 2014, which is also where he got his bachelor's degree. He received his PhD from UT Health Sciences and then did a postdoctoral fellow with Fernando Severo at McGill University, where he developed some of the first studies on how local control of protein synthesis within different parts of neurons is involved in the development of chronic pain disorders, definitely relevant to A2CPS and the work he's going to talk about today. He also spent time at the University of Arizona before coming back to UT Dallas. He's the recipient of multiple awards, including from the Rita Allen Foundation, the ISP, and serves as editor and reviewer for multiple journals. He's also an entrepreneur scientist. He and his group have launched a number of companies aimed at developing pain therapeutic products. And the work I think he's going to talk about today recently appeared on the cover of Science Translational Medicine. His talk is titled Molecular Characterization of the Human DRG and Its Implications for Pain Therapeutic Development. Join me in welcoming Dr. Ted Price.
Thank you, Stephanie. Can you all see my slides and hear me okay? All right, great. Yeah, I have an image to use on the first slide of my talks for the rest of my life now. So yeah, this is from the Science Translational Medicine paper, and I'll talk about that paper for a good part of the talk today. Thank you all so much for having me. I'm really happy to be here, and I'm looking forward to a discussion about how we can potentially work together using some of this data that we've generated. As Stephanie mentioned, I do have some conflicts. I will talk a little bit about some work that we're doing at Dolaromics, which is one of the companies that I've co-founded towards the end of the talk. This is our big group here at UTD. We have several labs that are working together, but we have our Center for Advanced Pain Studies that was co-founded by the three of us right here, myself, Greg Dusor, who I've worked with for almost my entire professional life, and also Michael Burton, who joined us and has a really booming neuroimmunology lab here in our group. And then we recently added Ben Kolber, as you just heard, and also have Kate Sadler joining us here in a couple months, which is really great for us. So, you know, if you're looking for faculty positions, continue to think about us, because we're going to continue to grow. And the work I'm going to talk about today was done by several postdoctoral fellows in my lab: Stephanie Shires, Deanna Tavares Ferreira, Pradipta Ray up here. And it's all been a collaborative effort of the whole group, and also a couple of people that aren't in the picture that I want to mention right now: Pat Doherty at MD Anderson and Rob Giroux at WashU. So I've been a little obsessed with species differences in DRG sensory neuron subtypes since I was a graduate student. Some of the first experiments that I did when I was a graduate student was looking at so-called peptidergic and non-peptidergic markers in different sensory ganglia, the dorsal root ganglion and the trigeminal ganglion in rats and in mice. And I was surprised to find when I was still a wee graduate student that while there's a very clear distinction between peptidergic and non-peptidergic markers in mice, that segregation of those populations breaks down a little bit in rats. And actually, we did a little bit of work that we never published when I was a graduate student using tissues from baboons. There was a large baboon colony in San Antonio. So Greg and I had the opportunity to recover trigeminal ganglion from some baboons. And this distinction was even greater when you looked in non-human primates. So this has been recognized by many people for quite some time, actually. So preceding the work that we did, Steve Hunt, who's an extraordinarily well-known giant of the pain field, this is a paper that he published in the Lancet in the late 80s. This is a not so great picture, but this is what stuff looked like back then in journals, right? This is from the Lancet. What they were doing here was looking at CGRP immunoreactivity in the human dorsal horn. They also happened to have dorsal horn from an intractable pain patient who happened to have died with this pain problem. And here you can see that CGRP, which is normally just found in lamina 1 in mice and also in rats, very clearly is labeling pretty much the entire substantia gelatinosa in both of these human samples. So we've developed this great collaboration with the Southwest Transplant Alliance, which is here in Dallas, that allows us to recover dorsal root ganglion and spinal cord, not trigeminal ganglion, DRG and spinal cord from organ donors on a pretty regular basis. So we have really great spinal cord and dorsal root ganglion samples, and we've been able to build on some of this initial data from people like Steve Hunt that's been around for a long time, and look at other markers. So here's a image from a published paper that Stephanie Chires was first author on in Journal of Comparative Neurology last year, where we see TRPV1, which in rodents is normally only found in a specific lamina within substantia gelatinosa, in fact, fills all of substantia gelatinosa in the human spinal cord. There's another interesting species difference here. In the human spinal cord, TRPV1 is also expressed by astrocytes, whereas in mouse spinal cord, I'm unaware of any data suggesting that TRPV1 is expressed in astrocytes. So clearly there's a major difference in the spinal cord innervation. And if you look in the DRG, you also can see that in mouse, there's only about a quarter or a third of neurons that express TRPV1 mRNA. It's the same as at the protein level. And in humans, we see that about 75% of human DRG neurons express TRPV1 mRNA. We've now also shown this with immunohistochemistry, and it's in this paper here, and then in the Science Translational Medicine, I'll show you the data in a little bit. In that paper, we did electrophysiology and also found that essentially all human nociceptors respond to capsaicin. So pretty substantial species difference there. So what we really wanted to do was to take this to the next level and move beyond just talking about species differences and really define what the subtypes of sensory neurons are at the single cell transcriptional level in the human dorsal root ganglion. And of course, this has been done in mice. It was done some years ago, and that data, I think, has really transformed what we can do in mice to pick apart circuits. And this is also happening in other areas. So in Yari's really great talk, you heard about how she can dissect these circuits in the amygdala, and single cell sequencing helps us to identify the neuronal populations that we can help to really, pick them apart at the circuit level using these amazing tools that we all now have access to in mice. So I think it's very important to understand this and where there are differences so that we can, translate these findings appropriately, use the knowledge base we have from rodents where we can do these amazing interventional studies and help us to advance translational science as much as we can. And then, eventually tools will probably develop where we can do these kind of manipulations in human tissues. So we're trying to set the foundation for that as well. So as I mentioned, we have this amazing collaboration with the Southwest Transplant Alliance. So I want to first shout out to the people at the STA that we work with. We used to work with their primary surgeon, Dr. Reese. He actually just retired about a month ago. We now work with their lead organ transplant surgeon, Dr. Funk, and Anna Cervantes, who's their lead surgical technician. So what we do is we go into the operating room with them after they're done removing all of the organs from the organ donor. And either Dr. Reese or Dr. Funk or Anna uses a surgical saw to open the spinal cord or the spinal column. And then we can remove the spinal cord and the DRGs. And you can see an immunohistochemical image of the spinal cord and the DRG with the dorsal root attached. And then in this picture here, you can see this is the DRG here. Hopefully you can see my laser pointer. And here's the DRG, which is really quite large in humans. This is probably an L4 DRG. The dorsal root going all the way up to the spinal cord. The spinal cord is actually kind of surprisingly small in humans. If you're used to working with rodents, the DRG is pretty small compared to the spinal cord. In humans, the DRG is quite large and the spinal cord is kind of surprisingly small. We're developing assays to do functional work. These include doing explants. You can see some explants here from DRGs. We're also developing spinal cord explants. And then we do dissociated cultures. So these are DRGs and dissociated culture. Oh, and here's the picture and video. This is the dorsal root that Stephanie's pointing out, coming all the way up to the spinal cord. So, you know, we have a prep if we can eventually start doing electrophysiology, which is one of our goals, where we could, you know, stimulate the dorsal root and record from spinal cord neurons, which we hope to do eventually. And if anybody develops collaborations with local transplant alliances, we're more than happy to come out and help to show you or the surgeons that are at the site how to do this. We're able to do this because Rob Giroux paid it forward and taught us how to do it, and you know, we're happy to do the same for anybody else that's interested. So feel free to contact me if you'd like. And okay, so what we ended up doing to do the transcriptomic work was using spatial sequencing. We used the Visium platform from 10x Genomics. There are now 2 papers on human DRG transcriptomics, one from Steve Davidson's group. They used a slightly different technique from 10x Genomics. That's the single cell sequencing on the Chromium platform. We chose this because these very large neurons in the human DRG are kind of hard to sequence using the 10x Chromium technique. Obviously, Steve and Nick Riva at NIH got this to work. But we had a lot of trouble with that and decided that we were going to do this spatial transcriptomic technique. And I have to say we were overwhelmingly pleased with how the data turned out from these experiments. So the way this works is you buy a slide from 10x Genomics. It has barcoded spots on all of these 4 tissue sections that or tissue areas here. And then you section the tissue that you want to put on these slides. You stain it so that you can see the cells. We use H&E staining. You image, and then you permeabilize the tissue. The mRNA binds to the barcodes that are on each one of these spots when you do that. And then you just build sequencing libraries, and you use those barcodes to tell you which spot the sequencing reads came from. So, you know, this allows you to combine the imaging with your sequencing, and you can see that many of these spots overlap with single neurons. So it's not true single cell sequencing, but it certainly approximates single neuron sequencing. And it has an advantage over NucSeq because we can sample the cytoplasmic RNAs, so we get a kind of fuller picture of the full cell transcriptome. And because things like satellite glial cells are found in all these barcodes, we can kind of filter out some of the signal that's coming from that cell type versus the neurons. So we use this information to identify 12 different clusters of human dorsal root ganglion neurons. We then divided them into some subclasses based on their cell size, which was super helpful because we could go back and look at the size of the neurons from the sequencing. And while human neurons are bigger than mouse neurons, you have the same thing. There's a distribution, and you can, I think, relatively easily spot proprioceptors and A beta neurons versus smaller C and A delta fibers. You can also do this in a blinded fashion to give yourself kind of a sanity check to make sure that, you know, your sequencing aligns with the cell sizes. So we identified 5 subtypes of A beta fibers, proprioceptors, and A delta low threshold mechanoreceptors, and then 7 subtypes of C fibers. And you can read all about it in the paper, so I'm not going to go into a ton of detail on that here, but I want to point out a couple of like key features. So one key feature is we've identified genes that do a fairly good job of distinguishing most of these cell types. So for instance, TRPM8, which we all know about because it was awarded the Nobel Prize last year. TRPM8 is activated by cold and is pretty specifically expressed by a subset of human neurons, which almost certainly are cold nociceptors. So, you know, TRPM8 is a really good marker for these because it's the receptor that's activated that transduces that cold signal.